There are open mics, and you can go towards them. And when we are done, you can find us in the crowd, and we can chat even more. So we have our first question. Hello, can you
It's working.
please introduce
Yeah.
yourself and tell which of us is the...
Sure. I'm Atal from India, and I have a question for you. It's very interesting and very exciting to see how, sensors can be designed and implanted to figure out these conditions. My question was regarding, the promiscuity that dopamine and, similar neurotransmitters like serotonin, they might also hinder the binding with the sensor, right, and identification. So how do you ensure that the sensor is specific to the neurotransmitter that you're looking at? And also the other question is regarding the implantation. Do you expect some sort of, issues with respect to compatibility of your sensor, and how do you tackle those?
Okay,
Thank you.
those are two very big questions, but about the interfering neurotransmitters. To be honest, when we are working with Alzheimer's and Parkinson's, they have very distinct patterns in how dopamine is in their brains. So they are either really high or really low. So this project was really started to detect dopamine, and then we kind of by accident found out that we can detect the subproduct of glutamate. So we have been focusing on these two yet, because the engineering of the nanoparticles should have been such that, first of all, they are sensitive towards these neurotransmitters. They can oxidize them, and at the same time, the interference, for example, ascorbic acid and uric acid were like the main issues for dopamine, because their electrochemistry peaks are basically in the same place. So once we kind of got that out of the way, we were really happy that we can at least detect dopamine. So we haven't really gone to see how the other neurotransmitters are responding on our platforms, but let's keep in touch, and when we do, I will let you know. Yeah. But about the implantation, we because we are a team of usually chemists and engineers. We don't have much clinical studies. That's why the last slide happened. We wanna kind of enter this phase, but we haven't, honestly. So
And
as soon as we can do that, I will let you know.
Apart from the compatibility, maybe, since the brain is so heterogeneous, where to
yeah, that's an
actually
issue, because we are trying to make the sensor very flexible. Correct. And at the same time, we are kind of experimenting with different types of carbon platforms. We have this idea, which will kind of start next week, that we have designed some very flexible platforms that we can kind of place the carbon on top of them and see if they can kind of stay intact once they are placed in the biological mediums. So that might be an answer. And we have been testing with something like inducing different potentials to see if we can combat biofouling. So that's another strategy we are using to see if we can increase the biocompatibility. Yeah. Right. Thank you so much.
Very exciting. Thank you.
Thank you. Hello.
Hi, I have a question for Nana.
Can you introduce yourself as well?
My name is Daphne. I'm a PhD student in biotechnology. Thank you for your presentation. I thought your project was very cool. I loved it. I was just curious to know about your rationale behind the choice of tentacles. Why did you choose to incorporate tentacles in your robot?
Yeah, thank you for the question. Octopus are really inspirational for self roboticists because their intelligence is distributed throughout their body. They don't have a single brain, if you would, yeah. So their intelligence is really distributed throughout their body, and they can do a lot of different tasks like manipulation of different objects. They can also crawl on the ground, and they can also, yeah, of course, swim. So this is why the octopus is really interesting for us. And the tentacle, it's a really simple structure, but it's able to produce thrust if we actuate it. So focusing on simplicity, this is why we chose to look at it.
Thank you.
Please.
Okay. Thank you for the nice presentations. I'm from Technical University of Munich, and I study electronics. So I find the water diet is very interesting. I have a question for Camilla. So do you have already some specific application using water diets? And what can we expect from it? Because you mentioned about you have networks actually formed by water diet. What can we expect from it?
Right, so thank you, great question. I was hoping somebody would ask that. So one application that we already tried was the networks that I showed. So it's an application not with the technology that becomes a device, but still we could visualize a mathematical theory that normally is just they just run numerical simulation. So this was cool to visualize a model. The other thing that we can use the diodes for, and what I did is the basic science work. So we are now working into implementing the diodes with actual applications. And one of them is, as I said, the liquid sort of computing. So creating, basic logic gates with, different designs, and then depending on so it would be with the flexible diodes. And then depending on the sequence of mechanical actuations that you perform, you get different, results, like in electrical, gating. And when I mentioned water harvesting, my vision, we're talking about 10, 15, 20 years from now, is an autonomous system that can, that reacts to external stimuli, for instance, humidity, so can detect when a plant needs watering, and then can, like the cacti that I said at the beginning, can harvest the water from the humidity and from the fog, and autonomously direct it to the plant that needs watering. That is my vision. In an arid area, obviously, we're talking about.
Thank you.
Thanks for the question.
Hi, my name is Inigo. I come from TUM as well. Very great presentations, a very great science, all of you. My question is for you, Bahar. I was wondering, because you mentioned also the glucose detectors, and that's very, kind of easy to implement. You just put it here and then you measure. How do you think that can be implemented for brain sensors, or those sensors that you have? Like, how will you collect the data? How will you measure all the results from inside? Because you cannot make a hole and put something.
Yeah, that's actually what we are going for. We want to do an implant.
Inside,
Yeah,
and
inside.
How do you collect all the data?
The gist of it is that these neurotransmitters will be released from the synapses. So you have like this very tiny space that is kind of concentrated with these nanoparticles. Neurotransmitters. And then if the sensor is tiny enough, for example, nowadays we have these microelectrodes that can be kind of placed in a way that can detect the molecules that are released in the synapses. So if we can kind of reduce the size of the sensor in a way that they can stay there. There are a bunch of groups that are doing this. I think one of them in MIT is actually really doing this. So they have live rats, and then they have placed a sensor in their brain, and they are the rats is living. But the problem is that it's a very short-lived period of time. So to best of my knowledge, the longest time that they could just let the sensor be inside is 154 days. I think I've seen just one more, but I'm not very sure about that study. But you can make the connections and you can have a living, breathing organisms or like, you know,
Yeah, but
animal.
how do you harvest the data from the sensor?
Yeah, and like
collect it?
the collection of data can be basically, well, for this purpose, it can be kind of transferred out via like some wireless technologies. But like if it's short term, you can always have it kind of stored in, and then you kind of put it out. Like in this case, for example, the rat dies, but then you will have the data of how these patterns are changing over time, and then you can maybe make assumptions on like if this is the case of how this pattern is, so maybe we can like know the brain in this regard better, because you are seeing the patterns but maybe not in real time. Again, one of the limitations with the things that we have today is that we cannot like monitor like 24/7 for like months and months and months. So yeah.
Thank you.
Hello, my name is Paris Rule. My question is for Dr. JD. I'm curious with like these cycles of these earthquakes, they're on the scale of years or like decades even. What's like if you're starting to see some plates that are generating a lot of stress, like they're in this time period of generating this friction over long periods of time, what's like the next point of like alarming the signal for people to leave or to evacuate the area? Or is it like, do we sit on the edge of our chair for the next 10 years of waiting for something to happen? Or
Yes, okay, thank you for that wonderful question. Yeah, and the stress accumulation for some faults even can last thousands of years. And once you study these kinds of faults, the uncertainties even become larger. And the main problem with this, with relying on stress, is actually we don't know the absolute state of stress of the Earth's crust everywhere. We don't know how it started. We don't know how much stress there is exactly right now on the faults. So it's hard to give a warning in advance to people. The best that we can do is give like a idea of a timeline of how often earthquakes can happen. That's why we call it like a recurrence interval. So we can't really still say, and probably not even in our lifetime, within the next decade or so, that there's going to be a big earthquake of such thing. So I think, yeah, that's one of the fundamental challenges in studying earthquakes, wherein the ground is opaque and you cannot see anything that's happening. It's not like a cloud over you. You can see the clouds coming in with the rain. So it's very indirect. And but yeah, if we can measure absolutely what the stress is in different parts of the Earth's crust, then perhaps we can move closer to that kind of projections.
thank you.
So we don't have to sit on the edge of our couches?
Well, I think here in Singapore, we're relatively safe from earthquakes.
Okay, cool.
Like to ask Ms. Bahar the question.
Hello.
Yeah, I treat intestine diseases, and it's really interesting to see your presentation on Parkinson's disease. Actually, one of the most common problems in Parkinson's disease is constipation, and sometimes these precede the clinical Parkinson's disease. We think it's because there's a decrease in the dopaminergic neurons in the enteric nervous system as well, not just in the CNS. So I want to ask whether your technique can be applied to the enteric nervous system. And I think it would be really interesting if it could, because it's easy to access the gut.
Well, to be honest, I'm not a doctor. I have no medical expertise. I'm basically doing electrochemistry, and I'm trying to make that sensor. So maybe it might be possible, but I have basically no idea about medical stuff. That's why we actually need clinicians in our group that can actually call these shots to see if, like, what if we can just put it somewhere else and detect the neurotransmitter there. So who knows? Yeah.
Okay, I guess if you're looking for some patients to put things into, yeah.
Sorry?
If you're looking for patients to put things into, please find us.
Actually, I'm really, really hoping that you find me on LinkedIn. And if not, you will go to the University of Turku Materials in Health Technology website and just shoot us an email. And I think we will be more than happy to arrange so many meetings with you. Thank you.
Thank you for your amazing presentations. My name is Sigrid and I come from Sweden. My question is for Ms. Camilla Sammartino. I'm curious about how you landed in using fleas. Since you opened your presentation with lizards and cacti, I am just a little curious about how you landed in wanting to like study fleas and their mechanisms.
Right, thanks.
I'm really sorry. Just we are out of time, so maybe we can
Wrap it up?
No, like you can answer, but like this is the last question.
Yeah, I'll be quick. So I was researching. Well, I'm not a biologist, so I'm a physicist by training, and I was researching, different organisms that have these diodes, that can spontaneously guide water unidirectionally. And I bumped into this study about fleas, and there was already an original, a study that tried to understand exactly how this organ in the flea works. So I have to give them the credit for the first, for finding it out first. And then we thought, okay, hang on, there's lots more that can be explored and can be done. And now we are exploring more animals, more insects, more plants to do more and more and more.
Thank you.
Thank you, everyone, for listening to our pitches. I hope you like them. And now we will leave the stage. Thank you.